Hi, this is Roger Green, host of the Surfing the NASH Tsunami podcast. This weekend, we are offering 5 conversations from Season 3, Episode 43, our look at the future of combination therapies, plus, from the more recent vault, a section of a May 2022 episode on NASH drug development that presages the topics we discuss in this week's episode. This week, our conversation from the vault comes from Season 3, Episode 25. Our look this past May at drug development. Conversation was led largely by Stephen Harrison and Jörn Schattenberg. This conversation starts with Louise Campbell asking whether design and management of ongoing trials will provide sufficient granularity on matching patient types to medications or drug classes. Stephen notes that we've not paid sufficient attention to this issue historically. In fact, he notes a range of key variables we do not explore at baseline. Genotype markers like PNPLA3, microbiome, and non-Caucasian population segments, to name 3. He also notes that some promising drugs have been killed because of trial design issues. In the end, he returns to some positive core concepts: combination therapies, looking for agents with multiple positive metabolic effects, and the importance of safety. At this point in the conversation, Stephen's transmission started to fail. Eventually, he left the conversation, and the rest of it was about cirrhosis. In some ways, the conversation we are having today picks up from these issues 3 months ago. So sit back, listen, enjoy, learn. When you're done, join the dialogue on our LinkedIn discussion group. Before we get started with the meat of today's podcast, one interesting note. We were fortunate to have Stephen with us this week. We were unfortunate in that about half the way through his part of the episode, the sound quality or the sound line or something started to punk out, so we got staticky, beepy, hissy kinds of things, and then eventually he fell off entirely. Please forgive the technical terms staticky, beepy, and hissy in that order. Mike is real good, Mike Wilson, our engineer, and fixes just about anything but couldn't solve some of this. So there will be points in this episode where I will interject and say, Stephen came in at this point and here was what he wanted to comment on. I will not be as complete, as eloquent or erudite as he is, but I'll get the major points made and we'll be able to catch the flow of what's really, I think, a pretty excellent episode. So it wasn't designed this way, but this might be a serious throwback episode for Surfing the NASH Tsunami. Stephen is with us today, which is great, and we have an extra-sode coming at the back of this episode from the Magical Pharmaceuticals presentation at March's CLDF Liver Connect meeting. So good afternoon, Louise.
Good afternoon. Nice to have Stephen back on the board.
And good afternoon, Stephen.
Yeah, hey guys. Good to be here.
Not only good to have Stephen back, but Stephen has what I think is probably the winning groundbreaker story about cross-culturation. So, Stephen, why don't you talk about bringing culture to Spain? And then Louise has to top that, and I have to top that. You're up.
Oh, sure. I've had an interesting past 2 weeks. After 4 jabs with the vaccine, I ended up getting COVID. So, fortunately, it was not a bad case. It did delay my trip to Europe, though, by about 4 days, but I eventually was able to test negative and fly to Spain, where I met some colleagues from Scandinavia who were introduced to Texas grilling. And this was interesting. So hopefully some of them will listen to the podcast. So I was able to, I would say a Weber barbecue grill was obtained locally, and I had the great pleasure of putting it together with a colleague. And then I was able to introduce Texas barbecue, and not barbecue in the sense of classic barbecue, but to use Louise's term, anything done out on the grill in Europe is barbecue. So we definitely applied a Texas grilling session to this meeting. So I was able to do hamburgers and steak. I brought a special seasoning with me from Texas, and it was an enjoyable time by all. So that was the introduction of Texas grilling to Scandinavian colleagues in Calpe, Spain. It was quite enjoyable, very fun. That was, I guess, my interesting tidbit for this episode.
I have to confess that for the year and a half our kids lived in Corpus, one of the things I loved best about Corpus was the best store-bought rubs I've ever seen in my life. Fish rubs, barbecue rubs, all kinds of rubs. So I don't even know which special rub you brought with you to Spain, but I can think of at least 7 or 8 off the top of my head that are better than anything we get back here, and people pride themselves on rubs in the East. So I'm sure you gave them quite an experience.
Well, it's amazing what that rub and butter will do to a steak.
Yeah, I was going to say, and for those of you cholesterol fans, Stephen did not know that he cooked with butter, but in fact he did that. The other thing that we famously do in the States is we take filet mignon and we wrap it in bacon, and then you put butter on top of that, so you get about as much fat as you can possibly get into this sucker. Yeah, that would be right.
And then maybe I'll just add professionally, we had a good week. I had a paper I've been working on for a number of months with an SGLT1/2 inhibitor. Finally, it was accepted in Nature Medicine, so that'll be forthcoming And that's nice to get that one across the finish line.
So we're waiting for papers from you now in Lancet and Nature Medicine. Anything else that's across the finish line but not yet on the silver screen?
Yeah, there's quite a few, but maybe in other journals like Journal of Hepatology. So still very, very good journals, but maybe not quite as challenging to get accepted.
And then, if I read it correctly, in the last week, Easel announced that one of the late breakers is going to be something you're presenting.
Yeah, we're very fortunate to be able to present the top-line data from the phase 3 Madrigal Maestro NAFLD 1 trial. So that'll be one of the late breaker presentations at Easel this year.
Excellent. So, Louise, go ahead.
Well, I suppose I can't compete with your omega-6 outdoor grilling story. Because that's what it would be if my husband didn't meat, although he...
Now, I didn't tell you about the omega-3s that were eaten as appetizers before. So actually our omega-3-6 ratio is okay at the end of the day.
Plus your saturated fat.
In fact, for Texan, it was dazzling, Louise.
Absolutely. It's the surfing NASH tsunami just on the recipes we're giving today. I can't compete on the cooking, although I was at the Ritz yesterday with my mother and my sister and our husbands and my dad, obviously, for my mother's 80th birthday. She'd requested to go back to the Ritz because she enjoys it so much. So we did that. So I assure you they do not external grill on the outside. It was all Michelin-starred food. And I believe the Queen was in the house earlier on in the day. So we keep missing each other. But professionally, it was nice.
Did she recognize you, Louise?
Well, the last time she saw me, I was in a red and white cape at Christmas, having just got married, and she was pointing out of the Range Rover. So we do keep missing each other. But that was several years ago. My near brushing with the Queen. And of course, it's the Jubilee this year. So we're ready for a party here in London. Professionally, it was nice to get back lecturing to nurse meetings. We had a meeting last week that was hosted by Dr. Falk, and it was nice to see so many nurses in a room together for the first time since the pandemic. So meetings are coming back, and we're able to get out there, which is great news.
That is good news. Jörn, you stepped in just in time to do your groundbreaker. You missed Stevens, but we'll fill you in later. It's got butter and lots of omega-6 and Texas grilling in it.
Yeah. By the way, I love that Texas food pyramid I saw the other day in Barcelona, Stephen, and you'll have to fill me on that one.
That was terrific. I love that.
I'm going to go with a personal. We did some sports this afternoon. I was running laps with my little one. It wasn't exactly running, but we stopped every meter and looked at something on the ground and picked this up and turned it around. Seeing the world around you in a certain slowness sometimes helps to realize that there's a lot of things to life, and that was very pleasant.
That's great. And my personal, in the tradition of what we always talk about here, comes from last Thursday night when we pretty well obliterated Arsenal in the North London Derby. Louise got to learn what the initials NLD stand for, which is North London Derby, which is Tottenham playing Arsenal. And right after this match, Arsenal plays Newcastle. We're hoping that Newcastle does better for Tottenham than they did for Liverpool. Because if they do, we'll play in the Champions League, and then you all get to listen to Louise and I bust each other about 2 matches a week all next fall and hopefully much of the spring.
When do you play Ted Lasso's team?
They just got promoted, so we won't be playing them until next season, and that will start in August. But, Stephen, it's funny you should mention that because Ted Lasso's team plays at Selhurst. What they call the dog track is actually Selhurst Park, which is where Crystal Palace plays. And Crystal Palace is Louise's second favorite team and the favorite team of her husband. So how did Tottenham do against Palace this year? I don't remember. We won what we should have lost, and we lost what we should have won, so I'm sure you beat us at least once, but I'll worry about that later. So with that, why don't we dive in? We lose Stephen in about 40 minutes. And what we realized is we've been doing lots of episodes this year. They've been extremely well received. In fact, to get ahead of the business report by about a sentence, we're going to hit 1,200 downloads again for what I think is the 8th or 9th week in a row, and really have elevated the platform for this podcast, which is exciting. But we've talked very, very little about drug development this year because there hasn't been a lot to talk about. One of the questions I got in Barcelona, and one of the questions I've started to get by email, is why aren't you guys talking more about drug development? So since we had Stephen with us this week, and Stephen is, in the words of Mazen Noureddin, the Duke of Drugs, this seemed like the perfect time to ask the question, what do we expect from drug development in 2022? And why haven't we talked about it more? So with that, I put together 3 questions that I will have up on my screen in just a second. But first one is just from where we sit right now, what does the pathway to drug approvals in NASH look like? And then the second is going to be, so what have we learned about that pathway so far this year? And what do we expect to learn the rest of this year? And if we can get through all that, I think this was a pretty good episode. Stephen, why don't I give you the floor first?
So the pathway to drug development in 2022 has not changed. It still requires a phase 3 trial showing histopathologic improvement. So a liver biopsy is still required, and the endpoints are the same. So NASH resolution without worsening of fibrosis, or fibrosis improvement without worsening of NASH. You could do both endpoints. You need to make the distinction between a co-primary and a dual primary. So Madrigal, in their press release, I'll use this as an example from last week, mentioned that they now are pulling fibrosis improvement up into a primary endpoint along with NASH resolution. That's a co-primary endpoint. It doesn't impact alpha. There's no alpha spend because you have an either-or situation. It's not a dual primary where you have to hit both. And I think that's an important distinction as well. But just the pathway to drug development in 2022 is very similar than what it was in 2021. There was a caveat that came out that was worth a brief little mention, and that is in the press release with Madrigal, it was noted that Madrigal had met with the FDA, and they actually decided to follow the FDA's lead in developing a phase 3 trial in well-compensated NASH cirrhosis patients. Now, what this does is it will be done in parallel with their phase 3 Maestro NASH program, which is the registrational trial. Now that trial, as you know, is fully enrolled with an expected readout in Q3 of this year. Their non-invasive trial, Maestro NAFLD-1, is also a phase 3 trial. They reported top-line data in a press release January 31st of this year, and I'll be presenting the top-line data at EASL in London next month. So you have the non-invasive phase 3, you have the registrational trial phase 3, and now there's going to be Maestro NASH Outcomes is the name of the trial, but it's in a cohort of around 700 or 800 well-controlled NASH cirrhotics, so well-compensated. What happens when you do that is you can swing your enrollment away from your Maestro NASH trial. In other words, historically, the idea is that you enroll about 1,000 patients in a registrational component of that trial, and then you continue to enroll about another 1,000 patients, and you follow all 2,000 of those patients or so out to an outcome, which could be 4, 5, 6 years. The idea being if you now switch to enrolling a well-compensated NASH cirrhotic trial, you follow those people to an outcome, and you still follow all the people you've enrolled in the registrational trial to an outcome, but you no longer have to continue to enroll in the registrational trial arm. So you shift to a population that is expected to hit an outcome sooner. So the idea being that it makes it a more efficient way to enroll phase 3 trials. So we'll see. This is the first time that's being done, and that probably is the biggest pivot we've seen in clinical trials in NASH in 2022, at least so far. So that was a bit of exciting and interesting news. And since it's been a while since we've really delved into where we are with drug development, maybe I'll give you just an update. When we look at the phase 2B paired histology trials that are either enrolling or fully enrolled in 2022. I'll just start with those that are fully enrolled. So there's Poxel's trial with PXL065, which is a mitochondrial pyruvate carrier modulator. That trial is fully enrolled. And if I'm not mistaken, I believe the press release from the company is that should read out sometime in the back half of this year, maybe as early as Q3. Then there is also other study that's fully enrolled, and that's with North Sea's icosapentate, which is a structurally engineered fatty acid. And that trial is expected to read out sometime late Q4 of this year. In addition, Akero has efruxifermin, which is an FGF21 agonist, and they have fully enrolled a phase 2B study in non-cirrhotic F2, F3 patients. And that should also read out in the back half of this year, I believe, also late Q3 or Q4. So from my perspective, those are the trials that we're expecting to hear from, the rather large phase 2B studies. There are many that are currently enrolling. Viking has a thyroid hormone receptor beta agonist that's enrolling. Sagimet has a FASN inhibitor. Excella has AXA1125, which is a combination of different amino acids. That trial is in phase 2B and enrolling. There's also 89 Bio, which has an FGF21 agonist that's enrolling. There is a DGAT2 inhibitor trial in phase 2b with Ionis. The drug is ION224. And then there's additional studies with GLP-1 GIPs. Lilly has a tirzepatide that is being looked at. Celgene has a J&K inhibitor that's enrolling. There's also lipocine with its testosterone prodrug. So that's kind of what is currently enrolling in phase 2b or has fully enrolled, and we're waiting on the outcome. So again, just in summary, 2022, we should be expecting to hear readouts from good-sized phase 2b trials. We should expect to hear something from Caro, from North Sea, from Poxel, and potentially others, but those I think for sure. Now, when we look at phase 3 and what potentially we're going to hear, as you know, there are 5 drugs currently in phase 3. Aramcol is on hold, so we won't talk about them anymore for right now. But resmetirom, I mentioned at the top of the podcast, we should be hearing from their registrational trial sometime in Q3 about their top-line results. Now, lanifibranor, which is a pan-PPAR agonist, PPAR alpha, delta, gamma, is currently underway enrolling their phase 3 trial. And semaglutide, a GLP-1 receptor agonist injectable, is also well underway enrolling their phase 3 trial. We will not hear about results from them this year because they are still enrolling, and it'll likely be 2024 before we hear from them. In fact, if we look at a timeline for surrogate endpoint analysis from semaglutide or from lanifibranor, probably going to be Q1 to Q2 of 2024 before we get that surrogate endpoint assessment. Now, we haven't given up on a beta-cholic acid. That's the FXR agonist that has already read out its subpart H approvable endpoint, met it on fibrosis with an 11% treatment effect delta. However, due to some therapeutic index concerns, that drug was not approved. They didn't stop the study. The study continued, so many more patients have made it to that pivotal liver biopsy, and we have many patients now who have been on drug for quite a number of years. And so it's hopeful that Intercept will reevaluate the additional data, both from an efficacy and safety perspective, and potentially refile. And that we're very hopeful might happen this year as well. So that's the phase 2b and phase 3 trials. I think it's worth noting some very key phase 2a trials. Now, these are early trials where we're really focused on safety and tolerability, but there usually is some study done to assess early efficacy, proof of concept, if you will. And these tend to be non-invasive endpoints. So we're not doing liver biopsies in these patients most of the time, but we're doing things like MRIs. We're looking at fibrosis biomarkers, ELF, ProC3. We're looking at liver chemistry test improvement, ALT, AST, etc. And so here we have a couple different FXR agonists. We have Terns 101 and Inyo, who have recently read out, and we're very hopeful that they will continue onward in their development. Hepion is focused on cyclophilin inhibition. It's a pan-cyclophilin inhibitor, and that has read out its non-invasive phase 2 and actually moving into a phase 2b trial later this year. We have High Tide, which is HTD 1801, which is a combination of berberine and ursodeoxycholic acid. They've read out their 2A as well, and we're waiting to see if they move into a paired liver biopsy phase 2b trial. Miricorilant, which is a mixed agonist-antagonist of the growth receptor and mineralocorticoid receptor, that is underway in phase 2a. And there's also another FGF21 agonist with Boston Pharmaceuticals. And then quite a bit of work being done right now with GLP-1 receptor agonist in combination with glucagon agonist or a GIP. There's AstraZeneca has one, Altimmune has one, Hanmi is another one. And then, of course, the galectin-3 inhibitor belapectin, which is an ongoing phase 2b3 trial. And that is a little bit different, that it uses an adaptive design, but the idea here being they want to prevent the development of esophageal varices. So that actually is the outcome being measured. So obviously to get into the trial, you can't have varices at baseline by endoscopy. And the the idea is with treatment, there would be less development of varices compared to the placebo group. So maybe I'll stop there and hear what you guys have to say, and maybe you could add some color to that. But that's kind of my perspective of where we are with drug development in 2022 as it currently stands on May 16th.
That's an impressive list. And of course, I said that when I commented on Stephen coming on in the Barcelona meeting. It's always a pleasure because he has all the oversight and then is able to recite most of these studies from the top of his head. Thank you for that summary, Stephen. And it refreshes my mind every time. My first question, maybe we go back to the FXR obeticholic acid because that's gone the farthest. And that means, you can reflect a little bit more, but we're seeing 48-month data for the first time in a phase 3 NASH trial. I think there's a lot of biopsies now being generated. So we're looking at 3 biopsies again, enrollment, the subpart H approval at week, 72, and then 48-month data and even endpoints coming out. Is the end of study biopsy even still important? Because we're really looking at endpoints, right? Or potentially also at biomarkers. But what's your take on that? How important is that long-term end of treatment biopsy? Because that's just the surrogate, right?
Well, yeah, except progression to cirrhosis is an endpoint. So the idea being with the 4-year liver biopsy is, in the placebo, we were expecting to see quite a number of F3s progress to F4s, which would be an endpoint. And that counts towards your overall number of endpoints. And so I think for that reason, it's important. However, at the end of the day, what's most critical is preventing decompensation. As you mentioned, this is a treasure trove of information to have this many patients, more than 1,000 patients, almost 2,000 patients, to progress through 72 weeks of treatment and have a follow-up liver biopsy. And I don't know how many patients have now made it to the 72-week point, but my guess is it's got to be more than 1,500. That's just a guess. And so with that amount of data being generated, there's surely quite a bit of safety data that goes along with that. And then, of course, you rightfully mentioned many of our patients have been on drug for, 4 years at least. So the safety data that goes along with that, you would hope that when you combine all that safety data with the additional efficacy data, that even if the treatment effect delta remained the same at 11% for fibrosis with no NASH resolution improvement, that given the large additional volume of safety data, that maybe that would be enough. We certainly could use an approval of obeticholic acid for the treatment of NASH fibrosis. I think we're all hopeful that they will refile, but there is no doubt there is a tremendous amount of data that has been collected. And let's not forget the phase 3 reverse trial in well-compensated cirrhotic patients with obeticholic acid. That trial has finished. Not only have they enrolled, but they've reached an endpoint, and we're just waiting for the top-line results from that trial. So that's a whole additional bit of safety data in a well-compensated F4 population as well.
Fully agree, and I think shows you the wealth. And of course, it's so important to carry the patients onwards. And I've seen that, to lose patients in that journey is or have them withdraw consent or not being able to collect those endpoints. That's something that's been difficult, but I think they did a good job. And most studies send us in keeping patients on drug, and we'll see that data emerge. And that's very exciting for me too.
Stephen, on what timeline do you expect we'll start to learn from the wealth of data? And what do you think will be the first things to see? I don't know how involved you are in the process, but your sense of what people are going to be learning and when I think would be really helpful.
Look, I think at EASL, the presentations are out. They're in the public domain. We know what's being presented. There's going to be, I think, some new data on non-invasive tests, particularly as it is related to resmetirom and THR betas. I'll be going through the top-line data from the Maestro-NAFLD-1 trial, which is 52 weeks of treatment. There's 2 drug treatment arms. There's an open-label arm and a placebo arm. And there is a incredible amount of non-invasive testing data that was collected, from FibroScan to MR elastography to MRI proton density fat fraction to other various and sundry wet biomarkers. So we will be mining that data long after EASL. There's no way we could get through all that data in 1,000 patients treated for 52 weeks in a 10-minute presentation. And you can't mine all that data in a matter of a month or two. So there will be many more very insightful pieces of data that I'm hopeful will be generated from that data set. Just look at the Stellar 3 and Stellar 4 trials with Gilead. My gosh, Jörn, how many papers have we published off of that data? And that was roughly 800 patients in Stellar 4 and probably the same number in Stellar 3. So 1,600 patients worth of data. So I'm very hopeful that we'll be able to mine that data for all the richness that it's likely to hold. And then in addition, there's probably going to be, I think, some very good looks at AI digital pathology relative to some more interesting concepts that are coming out focused around liver volume reduction and what that means to histopathology. And I'm anxious to see that. There will be more to come at AASLD as well. And so where we're headed, we're going to learn a lot more about mechanism of action of drugs. We're going to learn about how the non-invasive tests react to those mechanisms of action, because I think we all agree that not all mechanisms utilize the non-invasive test the same way. An example would be not all drugs move liver fat in the same way as others. And so maybe MRI-PDFF is good for some mechanisms, but not good for others. And we'll learn more about where that's applicable, I think.
And just to follow up what Stephen says, with the FXRs, we have 48-month data now, but with the Maestro NASH trial and the cirrhosis trial, there's just many more biomarkers being run in the back end. And I think the richness of data, as he said, is much deeper compared to the earlier phase 3 trials that we started, because back then, they didn't bother to put up all these NITs at the same time. So that's great, and I agree with that.
Yeah, my assessment of drug development in NASH is a lot like assessment of my own self when I look introspectively. And that is, you've heard me maybe say this before, I'm not who I used to be, and I'm not where I want to be. I'm a work in progress. And I think that's where we are in the field of NASH. We're clearly not where we used to be, and we're not where we want to be. But we're working our way through the challenges. We're learning from other failures. We're learning from success. And I think that it's all going to be better and better and better. Doesn't mean we won't have setbacks along the way, but I think learning from those setbacks helps us to understand how we need to evaluate these drugs moving forward, and we're getting better at it. Our pathologist colleagues are working hard to understand what's happening with treatment and understanding that resolution of disease is sometimes a tricky thing to understand histopathologically, but we're getting there. We're learning from that.
So it's a lot of stuff, right? That's a lot of data coming out from a couple of sources in a world that's really clamoring for a couple of things, which is A, first drug to market, and second of all, some realistic ability to understand more broadly what's going to happen here. Of all the various pieces of data that you've talked about, if you had to pick 2 or 3 that you'd consider tea leaves for the future of disease therapy?
Well, we'll just loop in Stephen every other week, and we'll get an update on this.
Yeah, failing that, though, people bet on futures. So I'm just kind of curious. If you had to pick 2 or 3, Stephen, that you think might be tea leaves for the rest of the year, what would be the ones you'd look at most, given that everybody thought OCA was getting approved 2 years ago, so prediction is hardly perfect?
Well, again, we want to learn from that experience. And the experience that I think we all learned, or that I personally learned, is that safety and tolerability are very, very important to the FDA. Maybe as much, if not more than efficacy. You got to be able to give a drug in NASH for the long haul, and it has to be well tolerated. It has to be safe. Maybe if we want to use the word tea leaf, I think, the top line results from the Maestro NAFLD-1 trial were insightful in that, you got 1,000 patients treated for 1 year, and it's very well tolerated and it's safe. So here you have a phase 3 trial reading out its primary endpoint is safety, and it hit that. The next thing is efficacy. And I don't have a crystal ball, so I can't predict the future, but I can say that I think half the battle has been won, and that is safety and tolerability. So if there is stat sig to be had on NASH resolution or fibrosis improvement, then I think that would bode well for the NASH community as a drug that is very likely to be approved. So that is something I'm looking forward to. The other thing I would say is when we look at the FGF21 class of drug, here we have a lot lot of early phase data suggesting that we're doing something very, very favorable in the liver. And maybe just as importantly, it's not just what we're doing in the liver, it's what we're doing extrahepatically. It's what we're doing to glycemic control, to atherogenic lipids, to liver fat content. So I think that's important, not only for the FGF21, but we see that on atherogenic lipids for the thyroid hormone receptor beta class of drug as well. The tea leaves that I'm going to mention now that are important is it's not just about what's happening histopathologically. It's what's happening holistically to our patients. Are they getting better? Are they reducing potential CV risk? Are we having an impact that is beyond just what we would expect to see in the liver? Because I feel like where we're headed in drug development in NASH is something that's bigger than just the liver. We would like to have a drug that impacts all the dysregulated energy metabolism that exists in our patients. Now, does that exist today? Well, I think we can clearly say there is not one drug that probably does everything we want it to do ideally. And that would be, we want to lose liver fat, we want to improve glycemic control, improve atherogenic lipids, lose weight, improve fibrosis, and resolve NASH. If we hit on all those, that's the holy grail. And I think it's early days to say that there is a drug that could do that. The GLP-1s are very, very attractive, but from the phase 2b trial that Phil Newsom published in New England Journal of Medicine, still lacking a little bit on fibrosis. Now, you can make the argument that they clearly showed that it prevented progression of fibrosis, but they didn't have a treatment effect delta on fibrosis resolution. Now, will they get there with larger numbers? Maybe. The placebo response rate was 33%, so that was quite high. In fact, I've heard arguments made, and I've made them myself, that the drug effect on fibrosis was similar to lanifibranor, but the placebo effect was different between the two trials, making lanifibranor positive for fibrosis, but semaglutide not.
After Stephen mentions the drugs and semaglutide, he notes that the GLP-1s are very attractive drug candidates and that he's anxious to see them in combination therapies, particularly to be used with glucagons or GIP agents. He mentioned that Altimmune has a glucagon GLP-1 receptor agonist combination, Hanmi has one, AstraZeneca does, Lilly does, and goes on to say that there are many that are looking at this not only from the obesity or diabetes perspective, but also as a NASH resolution agent.
So Stephen, can I ask you described such a wide-ranging sort of palette of drugs coming through, different mechanisms, which is wonderful to see because somewhere in there's got to be something that's successful. Do we run the risk, because you also alluded to the fact that everybody loses liver fat in different ways or can do the same as different people will get fatty liver for different reasons. Is there a risk with such a broad spectrum and the numbers required for trials that we miss the exact phenotypes of people who respond well to certain drugs because they may or may not be getting access to that mechanism? Do you think that runs the risk of losing medication that could be effective in other groups? Or is there a way that we can combat that?
No, you're absolutely right. I mean, we just tend to throw patients into the soup and hopefully at the end of the day the drugs have benefit, but we don't do a great job of precision medicine, of selecting patients for particular mechanisms of action. We don't right now, we as a general rule, we don't bring in genetic assessments of these patients. We don't look at PNPLA3 status. We don't look at HSD17 beta 13 status. We don't look at polygenic risk scores. We don't look at microbiomes at baseline. And we clearly know that what causes your NASH might be different than what causes mine, might be different than what causes Jörn's. There are multiple different mechanisms to get fat in the liver and multiple different mechanisms by which that fat can lead to lipotoxicity, downstream inflammatory effects which activate stellate cells, and vice versa. There are multiple different ways to shut them down, and there are multiple different ways by which there are redundant pathways to overcome a particular blockade. For instance, we learned with sinacrivaroc that you can block that, but maybe there are other redundant pathways that get around that. So to your point, yes, we need to get better at this. And I gave a talk at the CLDF meeting in Phoenix back in March, looking at the disparities in healthcare. And clearly, Hispanics are underrepresented in treatment trials. African Americans are way underrepresented in treatment trials. You can make the argument that African Americans seldom get NASH. That's true. They're the least likely ethnic group to get NASH. But when they get it, they can progress relatively rapidly. So we do need to develop drugs in that population. And there are very, very few data points points from which to point to there. So you're absolutely right. I mean, we saw it's not just that particular aspect, Louise, that troubles me. It's also, I think, good drugs are being put out to the graveyard because of the way they were read histopathologically. Look at MSDC0602K, a mitochondrial pyruvate carrier modulator that we published this paper in J. Hep et al. That the way the biopsies were read is non-traditional. So the traditional way is you read a baseline, you read an end of treatment, and then you look for differences between the two. But because we were concerned about temporal bias, we had the baseline biopsy reread in that trial. And when you reread those biopsies, the pathologist downgraded the baseline biopsy. I can't remember the exact number, but I think it was up to a quarter of the patients would have been excluded had that repeat baseline biopsy been read as the first biopsy. So obviously, the study did not meet its primary endpoint.
After Stephen noted that the study did not meet its primary endpoint, he said that when they went back and looked at it, as they did and reported in the paper, if you just took the baseline without comparing it to the end of treatment number, there was a statistically significant difference between the baseline and the end of treatment, post-treatment data, and it was consistent in a dose-response manner. So Stephen then went on to note that if that occurred, the drug would be in phase 3 trials right now if they had done what was typical as compared to what they thought was going to be this improvement. He finally went on back to Louise's point, and he noted that we need to improve our ability to select the right patients for the right trials, in part by learning how to interpret histopathology better. And he wraps up by noting that, as he says, and I quote, "Until the day we are looking at a non-invasive test, we need to continue to refine our methodology such that we don't leave good drugs behind."
So does that offer potential opportunities if you fail an FXR that you could enroll, that population may well then enroll into a GLP-1, for example, because they've already failed one mechanism, so we know that they're not responsive potentially. So you take that rich population that we've already tried and use a different mechanism, and we can work, I suppose it's selection, you naturally bring it out, and I know that would be an expensive way to do it, but it rolls into those other mechanisms.
Stephen provides a fairly rich, detailed response to Louise's question. He says, look, this is what combination therapy is all about. You can do sequential therapy. Let's say that 10 years down the line, you look through a crystal ball, we've got 3 or 4 different FDA-approved therapies. Each is monotherapy. They're not approved in combination yet. So you treat with the first one, you look at it 3-6 months down the line, you see what you've got. And maybe they haven't normalized their liver chemistry test. Maybe there's still some fat. Maybe their ProC3 or ELF or FAST or MAST scores or MEFIB or whatever haven't improved enough. So you need to pivot to a second drug. If they improve somewhat, add the drug. If not, maybe even replace. Hard to say. But you do that with a second drug. If that doesn't provide the necessary improvement, then a third, maybe even a fourth. And he says, ultimately, we've got to have these combinations to get at multiple different mechanisms within an individual liver synergistically. Because ultimately, one patient might be targeted by a mechanism or 2 or 3, and another patient might be targeted by a different mechanism or combination of mechanisms. What we want to do is to define the set of mechanisms for each individual patient that will serve that patient best, and then prescribe combination therapies to address each mechanism in that patient's set. Stephen concludes, I think that's where we'd like to be.
No, I mean, this is going to be difficult. We don't want to have patients fail and then enroll them in the next trial. So we need better NITs to predict that, maybe based on the MOA. And I think genetics could be one way. Stephen's detailed a lot of studies upcoming, but there are also some targeting certain genetic modifications, which I think could be very interesting because patients are selected based on the presence of these. And if you then target the therapeutic directly at it, I think that's something very interesting to me in the field here. And the other thing that we've seen, Stephen, is even if the FXR failed, then they're sometimes carried forward as a combination regimen. And it might be related to some of the, pitfalls you're mentioning, or, think about the aldafermin. We all think it showed effect on NITs that kind of moved the right way. Now, if you have a good combination partner, I would not say this is a drug that's ineffective, but it could be a great combination partner for something that addresses a complementary MOA pitching to then higher treatment responses. So again, as you mentioned, the failures lead the path forward, and we've learned many aspects, including, what we want to do better in reading out the primary endpoint of these trials, augmented with AI, for example. And as such, I know with all the data that's backing up and giving us support, I see this on a strong pathway forward.
Jörn, all that makes sense to me. One of the questions I was having was whether there is, in fact, a design or how we get to a design. As you commented, people fail on FXR, maybe go into a combination therapy. Who's pursuing that path, or Jörn or Stephen? How broadly is that kind of a path being pursued now? And do we have all of the tools, both in terms of the NITs and in terms of trial design theory, to be able to use that as a path to approval for a combination or monotherapy? Is that likely to happen? In response to my question, Stephen notes that we're not really there yet. We still have a ways to go. I mean, the idea is we have to get our first drug across the finish line before we can figure out how to enhance the use of that drug. Probably need 2 or 3 drugs across the finish line before we can think formally about combination therapy as a reality in practice. Now, having said that, Stephen points out, there are several companies who are acquiring or developing multiple assets in-house right now and beginning to look at combination therapy. Terns is a good example of that. Novo Nordisk is combining semaglutide with an FGF21 in one trial, and Gilead Novo Nordisk together are combining the FXR agonist cilofexor, the AAC inhibitor fracostat with semaglutide, which, as we've mentioned before, is a GLP-1 receptor agonist. Now, that trial has recently been published for advanced fibrosis, and they're starting a cirrhotic trial using the same combination now. In general, though, he concludes, most of the companies in late-stage development are monoasset companies like Madrigal, Inventiva, Akero, Bio89, others, Intercept with obeticholic acid. So he says they're really just trying to get their first drug across the line and get sales going so that they can improve their cash flow, their stock prices, etc. So right now, that's where most of the phase 3 companies are at. And until we get drugs and approval, he finishes, we're going to have to let the field sort itself out, but combination therapy is clearly where we're headed. I was going to ask you, we talked a lot last year about cirrhosis as a path to faster approval. And as we see studies going forward and people starting to look at things from a cirrhosis window, I wonder if you'd like to, Jörn, or both of you, would like to comment on how we see that as an approval path these days compared to, say, a year ago.
So from my perspective, there's clearly more activity in that arena. Stephen mentioned some of the trials. There's one on my way that we haven't discussed, which is a heat shock protein 47 and a very innovative drug delivery directly to the liver. And as such, I'm seeing more drug trials in that arena. Stephen mentioned the galectin, which looks at the prevention of varices. Some trials look at the regression of fibrosis, and taking these forward in more advanced disease stages or in more advanced clinical trial stages. The nice thing about it is we could get rid of biopsies because either, HVPG, which is not favored or also accepted as a surrogate, but we're looking at endpoints, preventable endpoints, prevention of the progression to significant varices or preventing the progression to decompensation, which I think is one way we do it. And as Stephen discussed, there's some phase 3 studies that are taking that along in parallel to their phase 3 non-cirrhotic trial. And this is something that came out of that FDA podcast that was, I believe, February 21, where they came up with the idea to develop that in parallel. And I think in the aftermath, we started to discuss this, and I'm seeing that in certain, I think it's picking up the cirrhotic treatment landscape.
What is the HVPG issue exactly?
HVPG is very closely correlated to outcomes, and it predicts future decompensation, bleeding, and mortality. So I think as such, it's a very strong test. The issue with HVPG is, of course, as if you'd like with biopsy, there's a certain variability to it, depending on who does the test. Is it well enough standardized? Now, it's well defined. So in an optimal setting, you're able to replicate these findings. But if you're starting with study centers that are unexperienced or do not have the training, you get more variability in there. And if I'm thinking back to the Emrickason studies, which used HVPG, we saw some of that variability at baseline and through the clinical trial. And as such, that's something that's always thought of being a little bit, a downside to using that test. On the other hand, it's even more than biopsy closely linked to clinical outcomes, which are highly relevant. And as such, they're still being pursued.
So let me let the audience know we lost Stephen at about 3-4 minutes ago, and I don't think he's going to come back in. Louise, comments, questions, thoughts to Jörn?
Getting a drug to market is important now for multiple reasons because it allows us to define the pathway. It allows us to see the interest and it allows us to get the education out. And once we set that landscape, any other medication coming through is going to be key. So unfortunately, we're obviously a couple of years behind with obeticholic acid not being approved a couple of years ago, but it's still likely to be the first one, I think it's been mentioned here, to be approved. And therefore, we can really start to write those pathways, the type of education, the type of people we're looking for. That will assist every other drug that comes into the marketplace and gets approved. I think the FDA are also keen throughout their study program on increasing equity and diversity, and have announced that you have to enroll people of different ethnic minorities. I think they've set some parameters on that. I think that was released in March of this year, and that's going to help some trials. It's probably going to be restrictive and require a little bit of thinking for other clinical trials to ensure that you get that population mix. But I think it's certainly looking encouraging from my perspective to obviously be able to drill down into the information that we're going to have to give to patients, members of the public, people about what the type of medications are, how they're going to be given, modes of actions, and how we put them into the real world. But we're not there yet. As I've said, a lot will come forward in NAFL pathways after we get the first drug, because we then will see that awareness build. And I already have conversations and people say, oh, why so-and-so, a friend of mine's been diagnosed with fatty liver. People are more aware of it in certain areas. So it is a common disease that now people are starting to become aware of. So let's see what the first drug to market does.
Thinking back of how Stephen started this session out, and I'll have to go back and listen to it myself again just to refresh on all the MOAs and companies he mentioned. The important thing is, even if obeticholic acid wasn't approved, you can achieve the endpoint of fibrosis regression in that time frame we're currently studying if you have a drug that hits the target, and obeticholic achieved that. Some people say, well, it's just 10% over placebo. My answer to that is fibrosis regression takes time. You cannot expect to roll back these fibrosis stages in all patients in these limited time frames. And as such, I'm very excited to see the 48-month data evolve. But bottom line is, it's doable. And, there's a lot of investors and companies sitting at the sideline. The good news is we can do it. We just need the right drug, and that will be emerging from that pipeline, I'm convinced. The path forward is a little bit more complex with the combinations, takes longer time and so on. But clearly in monotherapy, we've shown it before, and I think we're there and we'll show it again.
So, Jörn, it's interesting, the juxtaposition, and I think you just did that in 2 sentences a little more clearly than Stephen did, although he's certainly going in the same direction, which is if you take a look at the Maestro-NAFLD-1 data on safety and you take a look at the OCA data on efficacy, we've now proven that you can hit each half of that in the liver. And OCA not being, even if it wasn't deemed safe enough at that point in time, people have been on it for a long time, as you point out. And to the degree we've seen resmetirom efficacy data so far, we have reason to be hopeful. So we've proved we can hit each half of the equation, and there's a hope that either one of those drugs or both can swing both ways, number one. Number two, my guess is that in this market, combination therapy is going to mean vitality, because it's going to mean that either by acquiring multiple assets, as Stephen noted, Novo has done, Terns has done, others are doing, or by shaping partnerships, there will be lots of different ways for people to bring product to market. And that should keep research engaged, and it should keep people pushing towards an optimized drug therapy over time. So I think those are two, frankly, really encouraging signs. It would be nice if we could get one proof of concept that had everything in the same place, get it over the finish line. And at that point in time, one thing that happens, as we've all discussed on multiple occasions, is billions of dollars come into this market, both in terms of investment and in terms of education. And we start to make the medical community smarter, faster about, at least, if not what we do for well care, certainly what we do for sick care. And we develop drugs in wholesale. The question people ask me about that I've got no clue on, frankly, no real clue on, is exactly what the timeline is for all this. But I know the timeline for resmetirom would be anticipated to be sometime in 23. Lani, if I understand correctly, 24 if everything goes right. So we shouldn't be that far off. By the time we get to the end of the year, we should know whether the resmetirom data is going to be strong enough to look safe and efficacious at the same time. We thought we knew that with OCA, but I think we're a little chastened now. But that would be my sense, at least. Does that make sense to you two folks?
No, I agree with the timelines. I think, the other phase 3 we've discussed is semaglutide, and they're well on track and rolling. I'm thinking they're getting that conditional approval subcohort finished this year, and that will then read out down the line. And it's been quite a journey, and it's a little bit more to go, but they will be reporting out, and that's the good news. As
we set up our coverage for EASL, and even frankly whatever coverage we have Fifth Global NASH, one of the things we will want to do is look at those issues. At Fifth Global NASH, we'll have Louise and Rachel Zayas. They're interviewing people, among them drug company execs, about their studies. And if we send the right questions in the right direction, I think we'll be able to get some feedback from folks who are there. Madrigal will be there. Paxil will be there. Hepion will be there. I'm missing 3 or 4 of them. Please forgive. And then obviously EASL, everybody will be there. There's a late breaker on semaglutide and cirrhosis. There's a late breaker from Maestro-NAFLD-1 that they've already announced. And then there are all the abstract sessions. We'll start talking on this podcast in the next couple of weeks about how we're going to cover EASL. As soon as we figure figure out the logistics of doing that large a live event. For anyone who heard our NASH tag coverage, it's harder to do live than it is to do virtual. And the good news is we'll all be in London. The bad news is we'll all be in London. So it's going to be a little complicated, but we're looking forward to it.
If anyone, you can do it, Roger.
I hope so. I plan, let's put it this way. I don't only hope so, Jörn. I plan to. I just haven't figured out all the details yet. And thank you for your vote of confidence.
Let me loop back one more time. You mentioned the safety data and the efficacy data. I think both are important. The third pillar I'm seeing here is the quality of life data that we haven't routinely thrown into that equation yet. And there is some, even in the published domain now, some improvements with FXRs of certain energy and pain domains. And I think these type of, sometimes they're considered softer, but it's even an approvable endpoint. They're using validated quality of life tools. And now if you have a patient improve how he functions and you improve the surrogate of survival, and you maintain an acceptable safety profile, and you have three pillars that kind of point the right way. And I think throwing that into the equation will back up the drug development arena.
Boy, it certainly should, shouldn't it? I mean, if you can, quality of life is, as you point out correctly, what's unspoken here. We know that people with NAFLD, forget NASH, NAFLD, score lower on quality of life scales than others do. So you would think that if we can track that through the drug studies, that's a good thing. So in closing, just either take your pick, either the one thing that came out of today's conversation that surprised you or made you think about things a little differently, or if you don't have an answer to that, what's the one thing you're looking forward to the most in the rest of 2022 to tell us where all this is going?
I think I'll jump in. I think the one thing that surprised me, although I probably knew it, was the absolute breadth of the mechanisms and the method of action that these drugs are targeting across all of the drugs that are in investigation currently. Is just amazing. And I think the future is looking bright. And if I have to think a couple of years forward, I'm fairly sure we will have, as Jörn has said, and Stephen, that we'll have mechanisms now then in the real world that we'll be able to do. And I think going back to Jörn's point that he's just made, it takes me to Jose in Barcelona. Don't tell us what you want us to think. Tell us we'll tell you what we need. And a patient needs an improved quality of life. A patient needs to have a more stable life and stabilization of their disease. So bringing it back to what the patients have requested doesn't necessarily meet the endpoints currently set, but we're getting there. And I think that's the exciting part.
Thanks, Louise. Jörn?
Well, I guess Stephen's reflection on where he comes from, where he wants to be, and that he's in transition, which is true for most drugs, reminded me how we agreed on the endpoints now. And his opening statement was, we're still in the same arena. We have clearly defined approval pathways, and we're not starting to then all of a sudden read out biopsies a different way, because we've learned that it won't help to change the order, how they're read or reread the baseline therapy, and then throw out 1/4 of all the cases that have been enrolled. It's good to be reminded that we have clearly set a regulatory approval pathway. We have better defined how to assess those endpoints, with all the discussion around sampling variability, inability, or variability, and that really moved the field forward, I think. And then, in addition, the number of NITs we're doing today compared to earlier trials, with all the depth of knowledge emerging from that. And then, of course, the ability to link that to endpoints, which will eventually lead us beyond the biopsy. And that hasn't really come up that term yet, because we're not in the position to discard biopsy for accelerated approval yet, but we're on a good way based on all these studies that are ongoing and all the NITs that are being explored.
So Jörn, that was a good job of teeing up next week's episode, when we're hoping to have Quentin Anstey join us and talk about what happened at the recent Litmus event, entirely about NITs and moving that forward. The last few minutes of this conversation has, I think, made really an important point, right? Around quality of life. And that being, if you will, in some ways, the missing piece.
You mentioned Quentin and the European NAFLD registry. I mean, there's been a tremendous work the guy puts in there, and there's been strong data that emerged from the European investigators. And we've looked at some of that, the context of quality of life, and we actually were able to correlate lobular inflammation with decreased quality of life in these patients. Now, that hasn't been consistently reported from other cohorts, but be reminded that, again, the variability of assessment of inflammation could add to that. And bottom line, what it told me, it's a paper I think we published in 2019 in APT. The degree of inflammation in the liver impacts on the quality of life. And if we're able to change that, as Louise and Jose mentioned, this is what counts for the patient. It's that right upper quadrant pain if you have an inflamed liver. Stephen mentioned liver size at one point. It's the splenomegaly I see as a referral reasons. And I do see a certain correlation with that right upper quadrant, which kind of gives you the discomfort and the impaired quality of life. So that's all very closely linked. It's directly linked to the MOAs we're using to target inflammation. It's directly linked to pathophysiological changes. It's nothing the patient comes up with, I don't feel well, and we cannot grab that. So it's based on all the data we are seeing, very relevant, and that should improve. That's why I like those quality of life measures a lot.
Yeah, we agree. And this wouldn't be the first disease we've ever seen where an awful lot of stuff got coded as depression and turned out to be actually physiological in nature. So I think that's a wild card. I think it will also be extremely interesting, as Jörn points out, to see what the 48-month data shows on beta-carotic acid, because if a drug that was dinged for not being sufficiently safe carries patients 48 months without endpoint, without safety, without MACE or MALO endpoint of any significant note, then we start to, on the safety side, separate the hypothetical from the real. And I think that would be one way or another, I think that'll be a big plus. If it turns out that hypothetical turns into real, it will help us figure out how to do this better. If it turns out that hypothetical does not turn into real, it will make it easier to get things to the finish line. So with that, as I say, next week, two things will happen. Number one, we'll be back with Quentin. Number two, by next week, we will know what happened in English Premier Football, whether, in fact, Louise's beloved Liverpool won the championship or not, and whether my beloved Tottenham make it to the Champions League next year. So with all that excitement, folks, let me bid adieu to Louise and Jörn for the day. Bye-bye, folks.
Good evening, and I'll see you later.
Bye-bye.
I'll be back in a couple of minutes with the business report, and we will see you all later. Thanks. Bye-bye now.
And now for today's Extrasode.
Hi, this is Roger Green, executive producer and host of the Surfing the NASH Tsunami podcast. Last month, we attended and recorded presentations that Drs. Mazen Noureddin and Marcelo Kugelmass delivered, titled Emerging Concepts in Non-Alcoholic Steatohepatitis, or NASH. This presentation was developed and sponsored by Madrigal Pharmaceuticals. Today, I would like to share highlights of the two presentations, along with segments from interviews I conducted with Drs. Noureddin, Kugelmass, and Naim El-Khoury during the conference. Today, I would like to share highlights of the two presentations, along with segments from interviews I conducted with Drs. Noureddin, Kugelmass, and Naim El-Khoury during the conference. Please note, we have re-recorded some sections of Dr. Noureddin's presentation to improve clarity and eliminate background noise. The podcast represents the views of the speakers and does not necessarily represent the views of Madrigal. The content herein is for educational purposes only and should not be taken as medical advice. The first section of this podcast will include Dr. Noureddin's presentation, followed by our conversation that took place later that day. The second section will include Dr. Kugelmass's presentation, followed by our conversation that took place the following week. The last section is a brief discussion with Dr. El-Khoury, following up on some themes from the presentation. Let me take you back to the beginning of the presentation. If you wish to view the actual slides while following along, click the link that says Slides to view the presentation while listening.
Good afternoon, everyone. I'm Keith Miller from Madrigal Pharmaceuticals. We're pleased to support this program today on non-alcoholic steatohepatitis, presented by two people you know very well, Drs. Mazen Noureddin and Marcelo Kugelmass. We hope you find it to be an informative discussion and look forward to the discussion that it engenders. Mazen, the floor is yours.
So it's a true honor to be here today to talk about this pandemic and how things are emerging and the evolvement of our understanding of the disease, especially as hepatologists and gastroenterologists. I think all of us at one point realized how much we don't know. I mean, today's example of talking about disparities is like, it was really eye-opening. Like, we don't have enough studies. We don't have clinical trials in Asia and Middle East. There's one study on NITs. There are very few. And what I'm alluding to is basically like the mechanisms that came throughout the years, and as a hepatologist and gastroenterologist were eye-opening.
We're talking today about emerging concepts in non-alcoholic steatohepatitis. I would like to start talking about definitions, epidemiology, and comorbidities. Non-alcoholic fatty liver disease is a spectrum. Non-alcoholic fatty liver disease with the D is the entire spectrum. If you take out the D, you have this NAFL, which we used to call isolated steatosis or simple steatosis, and this is when you just have fat in the hepatocyte, in the liver cell, by 5% or more. The disease can then progress to non-alcoholic steatohepatitis, which is on top of the steatosis, you get ballooning and inflammation, and this is a cell injury. And this is a problem because that can lead to the more advanced forms, which is non-alcoholic steatohepatitis with fibrosis. The fibrosis can be mild, we call it F1, but the real category that we like to focus on is the F2 and F3, which is significant fibrosis. And this is really important concept in NASH. NASH with significant fibrosis, F2 and F3. And I want you to keep this in mind. Why? Because NASH and F2 and F3s is the group that correlates with morbidity and mortality. Of course, there's cirrhosis, and this is already too late, especially if it's decompensated. So again, I want to re-emphasize two things: the staging and grading. Grade point to steatosis, inflammation, and ballooning. And staging is also histology, but focus on fibrosis, F1, F2, F3, and F4, like similar to other liver diseases we have been dealing with. If we talk about prevalence, we have data now showing that the prevalence of non-alcoholic fatty liver disease is 25% of the population. And that can go up to 30% in South America and up to 31% in the Middle East. If your patients have type 2 diabetes, the prevalence of NAFLD go up to 50%. So now almost half of type 2 diabetic patients have non-alcoholic fatty liver disease. That's a big number. The other one that is very important is obesity. So 70 to 80% of obese patients have NAFLD, and up to 90% of morbid obese, they have NAFLD. Again, up to 90% of morbidly obese have NAFLD. And if you combine type 2 diabetes with obesity, the numbers will even go higher. I now want to list the common condition associated with NAFLD. And those are obesity, type 2 diabetes, dyslipidemia, metabolic syndrome, very important, and polycystic ovary syndrome. And metabolic syndrome, I just want to remind you what is it. There are multiple definitions, but the most common one used is if you hit 3 out of the following. You have to have 3 conditions, either obesity or waist circumference of 102 centimeters in men and 88 centimeters in women, 2 triglyceride of more than 150, 3 HDL less than 40 in men or 50 in women, 4 systolic blood pressure more than 130 or diastolic more than 85, or if the patient is on hypertensive medications, and 5 fasting glucose of 110 or greater. So if you had 3 out of 5, you're good. I also want to talk about other conditions that we usually don't pay attention to that is associated with NAFLD. 1, hypothyroidism, 2, hypopituitarism, 3, obstructive sleep apnea, 4, hypogonadism, and 5, psoriasis, the skin condition. I really want to re-emphasize on the bidirectional relationship between non-alcoholic fatty liver disease, type 2 diabetes, and metabolic syndrome. Why so? Because each one of them is standalone condition that make the other one worse. NAFLD, if a patient has NAFLD, they have a 2.25 times increased risk of type 2 diabetes. And indeed, if you have type 2 diabetes, that increases risk of NAFLD to get to fibrosis by 2 to 6 times. So they feed each other in an unpleasant way. They hurt each other. Also, type 2 diabetes is associated with cardiovascular disease and increase its risk by 0.5 to 2 times, as well as chronic kidney disease by 2.5 to 3 times. On the other hand, NAFLD has similar increased risk up to 2 times increased risk of cardiovascular disease and 1.5 times risk of increased chronic kidney disease. Finally, non-alcoholic fatty liver disease increases the risk of hepatocellular carcinoma by 10 to 100 times. And going back to non-alcoholic fatty liver disease in numbers, it affects 25% of the population. About 25% of those goes to NASH. About 25% of these NASH goes to cirrhosis. So now everyone is progressing. It's only 25% each time. And indeed, only minority go to hepatocellular carcinoma, about 1 to 4%. But what does that translate in terms of numbers? 25% of adults, they have NAFLD. 5 to 6% of adults have NASH, much less than 25%, but still a large number in the US. And 1 to 2% adult have cirrhosis of the US population. That's a problem. And there actually have been modeling study that looked into the projection of NASH over time. So a study published years ago showed that this number will increase by the NASH number by 63% between 2015 and 2030. 63%. For instance, the cirrhotics will increase from 1.3 to 3.5 million. The F3 will increase from 2 million to 4.5 million.
The F2 will increase from 3.4 million to 6.1 million. So here, if you take the F2 and F3, is going to be up from 5.4 million to up to 10.6 million people. That's a huge increase for the significant fibrosis of NASH patients.
Also, to re-emphasize on what's happening today, the data from our group showed NASH is the leading cause of transplantation on women. Other data show that NASH is the most growing cause of liver transplantation overall. And also NASH is one of the leading cause of increasing transplantation to the hepatocellular carcinoma. So a lot of things are increasing because of NASH. Most growing cause of transplant, most leading cause of transplant for HCC and in women. Problem after problem. Would like to switch gear and a very important concept with pathophysiology. And the hepatocyte usually is cell without a lot of fat. It's less than 5%.
But due to multiple factors like transportation of fat from the adipose tissue, the microbiome effects, the hepatocyte might get filled with triglycerides and other toxic fat, and that accumulation of 5% or higher leads to a very important concept, which is lipotoxicity.
Why lipotoxicity? What happens? The cells become ballooned, you start the apoptotic process, cell death, and there's a cascade of inflammations that leads to immune cell activation, activated sinusoidal endothelium, myofibrous blastic stellate cells, and you get reactive ductal cells. So that cascade of inflammation after the lipotoxicity is a problem. It's kind of wound healing process. But what that leads to usually is scar. That happens in the liver, you get fibrosis and eventually cirrhosis. I've been getting asked about the thyroid hormone signaling in the liver, and few hepatologists are new to this concept. However, this idea has been around since the 50s. Indeed, our endocrinologists know very well that thyroid hormone signal is very important for regulating cholesterol in the blood via the liver. And indeed, liver injury can impair this intrahepatic thyroid hormone signaling. And what that leads to is the accumulation of fat in the liver. And we'll talk in a little bit about that in more details. But I want to talk about that thyroid hormone pathway. So your thyroid gland secretes T4, and in the periphery, the T3 is the one that acts on cells.
So let's see what happened in the liver. You get the T4, and as I told you, the T3 is the active form. In a healthy liver, you have T4 getting converted to T3 via the adenosine 1. Also, there is some conversion via the adenosine 3 to the inactive T3. We don't want that as much. So you see that in healthy liver, it's not as much. What happens in the injured liver, it's the opposite. This adenosine 1 pathway becomes less relevant or less active versus going from the T4 to the inactive T3 becomes more prominent. And let's look now at the ratio, how we look at this impairment. If you have the T3 to RT3, which is the inactive, decreased, that means you're heading the wrong direction. So let's look in particular in the hepatic lipid metabolism and the role of thyroid hormones and thyroid hormone receptors. So here in the list, you have the functions of the thyroid hormones and the thyroid hormone receptors. De novo lipogenesis and synthesis. Let's talk about that for a second. You know that de novo lipogenesis is crucial for fatty liver. There's increase of that. So part of these pathways, you heard about enzymes like ACC and FASN. Well, there are data showing that the T3 and thyroid hormones regulate the ACC as well as the FASN. In terms of synthesis, the HMG coenzyme A, it's a major player and a major step in cholesterol metabolism. Indeed, it's thought that the thyroid hormones are one of the major regulators more than others for the HMG CoA. Let's move down. Fatty acid oxidation and mitophagy and mitochondrial biogenesis. So the TH-beta plays a role on the free fatty acid oxidation into the mitochondria through beta oxidation. So it's involved in that process. And finally, cholesterol metabolism. The liver takes cholesterol and try to convert it to bile acid, and we dump it through the bile. So thyroid hormones and thyroid hormone receptors regulate part of this process. The crucial steps that involves the pathogenesis of non-alcoholic fatty liver disease, such as de novo lipogenesis, fatty acid oxidation, the mitochondria, and cholesterol metabolism are affected and regulated by thyroid hormones and thyroid hormone receptors. And back to that, what you really want is the TH-beta to target lipid metabolism in the liver and not the TH-alpha.
Mazen, first of all, thanks for taking time out of what's already been an exceptionally busy day, and you have a lot left to do to talk with us. Congratulations. Very nice job. Very well presented. I do have a couple of questions for you. One, keeping in mind, I'm not a science guy. I think you did a nice job of describing what happens in the liver. But the part before that, where you were using your own body kind of as a way to explain where everything went and how things got from one place to the other, can you walk me through that?
Sure. Roger, it's always a pleasure to be here. One of the questions that came recently from a lot of hepatologists and gastroenterologists, why we're talking about thyroid hormone receptors, thyroid hormones in general, and the relation to the liver. As I said, it's a very known relationship since the 1930s that thyroid hormones are crucial in regulating cholesterol levels and lipid metabolism in the serum and in the liver. Going a little bit back to the basics, the thyroid is in your neck. It secretes T4.
The thyroid gland is regulated by something higher, the pituitary gland, which secretes thyroid-stimulating hormone, or TSH, and the pituitary gland is regulated in the brain by the hypothalamus.
The T3, which is the active form of thyroid hormones, works in the cell, and there are receptors that are involved. Receptors of TH alpha, which is called THR for abbreviation alpha, and THR beta. And as you know, receptors are involved in multiple parts of the body, and sometimes they exist in certain part of the body and not another part. And the part that THR alpha are in is the heart and bone. THR beta exists in the liver and the kidneys.
Is T3 metabolized in all those places or just in the liver?
You get T3 in all these places. It depends on the receptor and all that. But what happens in the liver in particular, the T4 through the adenosine 1 goes to T3, the active form. Some of it through the adenosine 3 to the inactive form of T3. And that's what you always want, to get the active form, the T3. You wanted to do the work. You wanted to metabolize the lipids in the liver and keep doing its job. In unhealthy liver, you have less T3. You get more inactive T3 or T3. There's beyond that the lipid metabolism effects of the thyroid hormones.
Right.
As I said, I'm going to summarize them and I'm not talk about every single level, but it is de novo lipogenesis and synthesis. So they play thyroid hormone and thyroid hormone receptors play a major role. So we talked about de novo lipogenesis and synthesis. 2, they play a role in free fatty acid oxidation through the mitochondria via a process called beta oxidation, which is crucial for NAFLD and NASH. And finally, the metabolism of cholesterol in the liver. Cholesterol comes in the liver, needs to be converted into bile acid, and it gets dumped in the bile. The thyroid hormones as well, the thyroid hormone receptor play a role in that as well. And finally, there's gluten metabolism regulation as well. It's really in the heart of the entire process of NASH. And as I mentioned, one of the major enzymes, HMG coenzyme, is regulated by multiple things. And that's include insulin, glucose, estrogen, and thyroid hormones. And there are data showing that thyroid hormones actually play the major role in its regulation.
If I recall correctly, HMG-CoA is what statins regulate, right? That makes complete sense to me because we all learned 30 years ago that we thought antihistamines were the things that stopped you from sneezing until we found out that H2 was the thing in your stomach that gave you ulcers. So why is a thyroid hormone in the liver? The answer is why is histamine in the stomach? Things wind up where they wind up. Here's the other question. You did a really nice job of demonstrating all the relationships that exist between various metabolic diseases.
This is key because it's a metabolic disease, non-alcoholic fatty liver, and it's tightly connected with type 2 diabetes, metabolic syndrome. And indeed, if you look at the two diseases, with non-alcoholic fatty liver disease, there's higher association as well as future increased risk of type 2 diabetes. The association with NAFLD and type 2 diabetes is up to 2.2 times. The other way around, if you have type 2 diabetes and metabolic syndrome, you're more likely to have risk of fibrosis in non-alcoholic fatty liver patients.
Are there any causal relationships in this?
It's mostly associations, and the underlying mechanisms as a cause are being studied. I'll give you an example. The inflammatory status in the body. We know that NAFLD can cause that by itself. And if you have plaques in the heart, you don't want that systemic inflammation. So it makes things worse, right? Same thing for obesity and type 2 diabetes. It's inflammatory. This is just an example. Inflammatory state that can lead to increase the risk. I'm talking about NAFLD here in particular. The direct mechanisms to cause a heart disease as isolated risk factors are still being investigated.
Because the underlying question I would have if I were a doctor is, hey, I know how to treat diabetes. I know how to get glycohemoglobin down. I know how to treat cholesterol. I'll get you stand down. I know what to do about obesity. So if I'm doing all those things, why do I need to do this?
Data has shown over and over that NAFLD will come in univariate and multivariate analyses as a risk factor by itself, for instance, for cardiovascular disease. So we can say that based on univariate and multivariate analysis, the non-alcoholic fatty liver disease coming out as a risk factor, and thus it's suggested it has a cause and leads to cardiovascular disease.
These are not merely a correlation matrix, but if you try to boil it down, that NAFLD pops out as predictive of the likelihood of having cardiovascular disease.
I just want to mention one thing back. We talked extensively today about how the lipids are regulated in the liver through different mechanisms. It happened when we talked about TH and THRs, beta and alpha, or beta in particular for the liver. So it's plausible that you think that the liver is causing the heart disease through this abnormal or normal lipid metabolism, because we know the whole thing starts from lipid and inflammation. So that's important. And the liver is main player and a major factory for regulating all that lipids and glucose metabolism.
And now let's hear from Dr. Marcelo Kugelmass.
Hi, everyone. Thank you, Mazen. And thank you to Madrigal for sponsoring this program. We're going to finish the talk. Let's start talking about morbidity and mortality. NASH with fibrosis is associated with an increased rate of mortality. As you go from simple steatosis to NASH without fibrosis to non-cirrhotic fibrosis and then to cirrhosis, that is when you have all the effect of this disease. Now, Mazen explained that for liver outcomes, the people that are at risk for liver outcomes are those with stage 2, stage 3, and stage 4 disease. And I am concerned that most of the guidelines talk about, well, the other ones will not have liver outcomes, but they will have other kinds of outcomes. They will still have cardiovascular outcomes, and they will still have non-hepatic malignancies and things that need to be looked at. So the fact that they don't have advanced liver disease doesn't mean that we have to give them up and that they are okay to go home and never come back. The other issue is that this is a progressive disease. So today they may be okay, but I want to evaluate these people maybe on a yearly basis, maybe every 2 years, maybe every 3 years, but not give them a free pass, don't ever come back, because the disease could progress. And as somebody who has experience with laboratory testing and non-invasive testing, people make mistakes all the time. So if you think that clinically what you're seeing is different than what a single test tells you, repeat the test. If the patient has early fibrosis, they're okay today, they have other things to worry about, but bring them tomorrow and reevaluate them. The risk of liver-related death is statistically higher only after progression to stage 2 disease or higher. For stage 2 disease, the rate ratio is 9.57 compared to 1.41 in stage 1. And then you go to 16.69 and 42.3 for cirrhotics. You see among patients with NASH, those with cirrhosis are at greater risk for decompensation, hepatocellular carcinoma, or death compared with less advanced fibrosis. So you have stage 3 on the left, stage 4 with compensated disease, and then stage 4 cirrhotic, still with compensated disease by child-to-cot Pew scoring, but they are already showing one extra point. They have progressed in some way. And so those could be clinically translated to the beginning of portal hypertension, for example, which makes a difference between cirrhosis without or with portal hypertension. So overall mortality or liver transplantation, 3% for stage 3, and for stage 4 is 11% for the more advanced stage 4 or the beginning of decompensation, which is not quite decompensated yet, 58%. Same for the first occurrence of a major clinical event, whether it's hepatic decompensation, it's mostly hepatic decompensation. The liver cancer, non-hepatic malignant neoplasms, and major vascular events are similar in between stage 4 with an A5 child-to-cot Pew score or A6. And because of sample size, there were none seen for non-hepatic malignancies or major vascular events in the A6 group. This is older data from Paul Angulo, and we've all quoted this paper multiple times. Cardiovascular disease is being the number one cause of death for this population, 38.3%, non-liver cancer, 18.7%, and then the third most common cause is liver cirrhosis and its complications, 7.8%. Now, the change in disease activity in the NAS score is associated with changes in fibrosis. In other words, inflammation is what drives this condition to continue to deposit fibrosis, alter the structure of the liver, and alter its function, leading to cirrhosis and the complications from cirrhosis. So a high NAS score at baseline has been associated with progression to fibrosis stage 3 and 4. So in other words, if we were to do liver biopsies on most people, and I definitely do not advocate this, and we shouldn't really, if we saw those with higher NAS scores, with more ballooning and inflammation in their liver biopsies, those are the patients that deserve the closer follow-up. And if we had a way of assessing that inflammatory component and a non-invasive test, that's what we would like to follow. Now, there's technologies being developed for that, not part of the purview of today's talk, but there's people that are working on this to try to differentiate what is NAFLD or NASH with or without different intensity of fibrosis. And then the trajectory of this fibrosis change is directly associated with changes in disease activity. So that's what we want to modify. Patient identification. This is simple. If you see a patient, 1 out of every 4 will have non-alcoholic fatty liver disease. And if you see somebody with diabetes, half of them are going to have non-alcoholic fatty liver disease. So my best way of identifying new patients is to go to the endoscopy suite, go through the past medical history and order FibroScans for everybody with diabetes. It's simple. So what are the things that we can do for these patients? Simple evaluation scores, blood work or wet biomarkers. FIB4, probably the most commonly used. The NAFLD fibrosis score and the APRI are utilized probably less. They have less significant data behind them. FIB4 is part of the guidelines. Easy to obtain. There's an online calculator. You can pretty much do it for anybody. Then dry biomarkers, the imaging techniques. Typical tool that primary care will use and send you a patient for. Patient went in for whatever reason. They were accompanying a family member that had an emergency, went through the emergency room, and they got a CT scan themselves as well, or an ultrasound. And the ultrasound showed fatty liver, the CT scan showed fatty liver, and then the patient gets referred to us. Much less commonly, people will get MRIs, and definitely we can do MRI with PDFF, with proton density fat fractionation, to quantitate the amount of fat in the liver, and then MR elastography. Now, MR technology, while it might be the best test there is out there, is usually the least one utilized, simply a matter of access and cost. So conventional ultrasound and CT scan tend to be the most common imaging techniques. For many of us, we have access to vibration-controlled transient elastography or FibroScan, and there are several commercial companies that have machines for this. It is a very helpful test. It's non-invasive. It doesn't hurt. There is no risk, and it's incredibly cheap. It's very, very good for its negative predictive value. From a hepatology point of view, if your VCTE shows that the patient has no significant stiffness to the liver, then that liver is likely squishy and healthy, and that's a good thing. If the FibroScan tells you this liver looks stiff, the only meaning of that is that this patient deserves further workup. So the high kilopascal result for stiffness in the FibroScan just means you need to do further workup for this patient. Lastly, proprietary serum testing, and mostly it would be the ELF. PRO-C3 and NIS4 have less data behind it. The ELF is recently approved by the FDA. I don't know how many of us are using it, but we will probably use it extensively. It'll be also a little bit different from the FIB4 in that the FIB4 doesn't cost more to the patient. The ELF will. How often should we do this testing? I think that is a great question. Most of the data that we have is about the initial evaluation of the patient. There's not a whole lot of data looking at repeating these tests yearly, every other year, according to stage. How should we do it? There's not a whole lot of guidance. But if you have a patient, the more advanced the disease, the more you should think about repeating the assessment. Fat accumulation tends to change, and that is something that you can follow, and it's quite dynamic. You could follow it on a yearly basis or even more frequently. Fibrosis tends to be a lot slower, and the initial data would say that you don't want to look at fibrosis every single year because you're going to do a whole lot of unnecessary testing. There's a couple of screening algorithms out there. This is one from AGA, and we start here: primary care, endocrinology, gastroenterology, and obesity specialists. The knowledge depth of each one of these groups is probably different, and the interest of each one of these groups is probably different. So depending on how well versed they are, they want to go through part or whole of the algorithm. As I said, step one is identify the patients at risks. You can really start by looking at them. So 2 or more of the metabolic risk factors: central obesity, dyslipidemia, hypertension, insulin resistance, or diabetes. Then everybody with type 2 diabetes, should be evaluated for fatty liver disease, and anybody that shows steatosis on any imaging modality, or anybody with chronically elevated liver tests, because NAFLD is now the number one cause for chronically elevated liver tests. Step 2 is actually try to differentiate what is causing this condition. We send a test of all the liver serologies, we do imaging, and we assess for autoimmune conditions, viral hepatitis, inherited disorders, disorders of iron metabolism, whatever it is. We do ask them about alcohol, and some of them will actually tell us the truth. The patients with intermediate risk or with high risk, intermediate being 8 to 12 kPas and high risk greater than 12 kPas, those require further testing. The AGA guidelines has referred to a hepatology for liver biopsy or MR elastography or monitoring with reevaluation of risk in 2 to 3 years. My editorial comments. If you're in the yellow category, I don't necessarily do a liver biopsy unless there's a differential diagnosis to be elucidated. If I have a suspicion that they could have autoimmune hepatitis or something else, I'll do a liver biopsy. Otherwise, I will not do a liver biopsy. I will try to get them into a research study through which they will always get a liver biopsy. But I will not do a liver biopsy for somebody that I can follow in the clinic. I think it's just too invasive and not necessarily a good test. It has a lot of variability, both inter and intra-observer variability, and a lot of sampling error. So I'm not a fan of liver biopsies. So you get what you can. In my practice, it'll be VCTE. And for the patient that could have advanced liver disease, I will probably want to do another dry NIT. In this case, I will push for some sort of MRI. I will still stay away from liver biopsies. I don't do liver biopsies to prove that a patient has cirrhosis. I think that we're beyond that in hepatology. This is the recommendations of the Diabetes Society, and what I want you to focus on are patients with type 2 diabetes or pre-diabetes and elevated liver enzymes, the ALT in particular, or fatty liver on an ultrasound should be evaluated for the presence of non-alcoholic steatohepatitis and liver fibrosis. Evidence level C, but that is a recommendation for diabetologists. These are the EASL guidelines, and they're similar. So patients at risk for chronic liver disease, check history, rule out all the different conditions with the other usual workup. If you find that they have, let's say, autoimmune hepatitis, you refer to the specialist that manages that particular condition. And then if it's fatty liver disease, you decide whether it's alcoholic fatty liver disease, non-alcoholic fatty liver disease, or both, which coexist in many patients. Do a FIB4. If the FIB4 is low, less than 1.30, that patient is done for the hepatology workup, but their care continues for all the other conditions that they have. And if they have FIB4 greater than 1.3, which is the indeterminate and high results, they will go to liver stiffness by transient elastography. And then you use the ELF or other patented serum tests when available, and you make a decision whether they need a liver biopsy or not, and what kind of intervention you're going to do. The most common side effect of chronic liver disease, the most common symptom of chronic liver disease, everybody that ever treated somebody with hepatitis C knows the answer, is to have no symptoms. Clearly, many of these patients will be obese, many of them will have sleeping problems, obstructive sleep apnea and things like that. But the most common symptom is to feel fine, which is actually sort of like a problem from a provider point of view, because if you feel fine, you're asking me to do all these things for what? I feel fine. I don't want to change. How are we asking them to change? Well, these are the guidelines. Because we have no approved medication at this time, lifestyle and dietary modification is the way to go. I give them a single sheet of paper that has on a single face all the dietary and lifestyle recommendations, because nobody ever turns the page. So the instructions need to be simple. You need to be able to put them on the refrigerator, and then you might have a chance to follow them. Believe me, I have had at least 3 patients that have lost over 100 pounds just with dietary and lifestyle modification. And I have had about 100,000 patients that didn't lose any weight. So while this is effective, it's not necessarily effective for all. It's just effective for those who are committed to doing the right things. So energy restriction, and energy restriction starts by just eating less. I eat 3 servings of everything. Energy restriction for me would be 2 servings. My wife eats out of a normal dinner plate. Energy restriction for her would be to eat out of a salad plate. Give the patient something that they can actually follow. If you have access to a dietitian, if the patient can see a dietitian, if they can see somebody that will actually give them guidelines, coffee consumption. Coffee is fantastic. It makes the liver better. How many cups of coffee? 4 cups of coffee if they can get to it. Don't go from 0 to 4. in 1 week because you're going to end up in the ER with tachycardia, sweating, and hypertension. Go slowly. And if they don't want to get there, okay, they don't get there. But if they want, coffee is good. Which coffee is better? The cheapest coffee that has caffeine is the best. Macronutrient composition. Less fat, less carbohydrates, and reduce fructose intake. Then control daily alcohol intake. If you have liver disease, no alcohol is better than any alcohol. If you want to get into a lengthy discussion about how much alcohol you should have, that means that you drink too much. So my standard recommendation is don't drink any alcohol. If you have a child that gets married, yes, of course you can toast at their wedding. If you have 100 children that get married weekly, that's a problem, okay? Increase physical activity. Walk, run, row, treadmill, elliptical, whatever it is you want, but do something and sweat at it. But because cardiovascular disease is the number one cause of death for these patients, make sure your ticker is okay. So if you have heart disease, go see your cardiologist first. So as I said, there are no currently FDA-approved therapies for NASH, and so lifestyle and dietary modification are still the cornerstone of treatment. And if you belong to a practice that has access to research, that is the way to go. Bariatric surgery is excellent, and it has improved basically all outcomes of these conditions. Bariatric surgery, unfortunately, changes your lifestyle for good, and some people actually learn how to beat it. So not everybody's a good candidate for bariatric surgery, but when it works, it is the single thing that works the best for these patients.
Marcelo, thanks for taking time in your incredibly busy office schedule today for this conversation. Let's dive right in. In your mind, what makes it important that we be treating NASH now as compared to diabetes, obesity, whatever else is in that constellation of diseases?
NASH will become the third most common cause of death, or has been shown to be the third most common cause of death for this group in general, after cardiovascular and oncologic causes for mortality. This is already the number one cause for all chronic liver disease, for all cirrhosis, for all liver cancer, and for all referrals for liver transplantation in the United States. For most of us who believe in preventative medicine, we would like to treat now and as soon and as well as possible so that we can prevent some of these negative outcomes in the near future to come.
So today, how do you treat your diabetic patients? What does the initial conversation look like? How do you frame that?
I try to keep it simple. Basically, I tell them that everybody with diabetes is at risk of developing fatty liver disease, and that a proportion of those patients with diabetes be screened for this condition in endocrinology offices. They would probably get blood work to include liver tests from time to time, as well as possibly a liver ultrasound. In our practice, we will use that blood work that they have available to calculate FIB4 index. If they are at intermediate or high risk, we will progress on to doing a fibroscan. So we're lucky enough to have one of these vibration-controlled transient elastography technologies. It is a good tool to screen these patients. If they show a high result on the VCTE, then we go on to further testing. So the pathway is fairly simple. They tend to come with labs that can already tell us whether they have a suggestion on these wet biomarkers, and then we use a dry biomarker in my office to do that. And again, this technology is interesting because it can rule a lot of people out. It is not invasive, not painful. It is readily accessible to the patients. It tends to be cheap for everybody, and it definitely has no risk to doing it.
Naeem, I want to take a couple of minutes and talk a little bit about the San Antonio study. Could you just walk through a little bit what you folks did and how you collected data and how you got to FibroScan people, really, and then what the results were?
Yeah, absolutely. I mean, this was really the largest prospective study to evaluate the prevalence of both NAFLD and NASH. The way the study was designed was individuals coming to the Army Hospital in San Antonio for their screening colonoscopy. So it was mainly middle-aged adults in their 50s. They had no idea they had any liver disease. So they had no history of hepatitis C or NASH. They didn't have significant alcohol consumption, and they didn't have any known history of elevated liver enzymes. When they came for the colonoscopy class that we do before they undergo the colonoscopy procedure, we offered them to learn more about their liver health. And if they said yes, we signed them up for the study, they signed the consent, and then we actually took them to get FibroScan, but also MRI PDFF and MRI elastography biopsy, and this was the initial assessment that we did. We use MRI-PDFF to quantify liver fat. We also had the criteria related to transient elastography and MR elastography and corrected T1. So if they're above a certain threshold, we also offer them a liver biopsy. What we found is that out of 664 patients that signed up for the study, that based on MRI-PDFF, 38% had evidence of NAFLD. Then we offered them the biopsy, and we found that 14% had evidence of NASH on liver biopsy.
A couple of questions. If I recall correctly, only about 80% of patients that you offered the biopsy to took it. Do I recall that number correctly?
Yes. So we had, some patients that actually declined to have the liver biopsy, which is understandable.
Certainly. Is the 14% number corrected in any way for those who didn't have the liver biopsy? Or if you didn't take the liver biopsy, did that automatically put you into the non-NASH group?
Yeah, so if you didn't have the biopsy, we assumed that you don't have NASH. So actually the 14% that we came up with could be an underestimate of the true prevalence of NASH.
I'm not sure you captured this number, and if you didn't, I'd just love for you to guess. How many of the people that were in the colonoscopy class that you offered to learn about their liver said no?
Honestly, the majority of people said yes. So if I remember the numbers correctly, we approached about 850 individuals and about 660 said yes. So the majority, about 75%, said yes.
I remember a really high percentage of the patients that you identified that had NAFLD in the San Antonio study also had diabetes. What was that number?
The entire population, it was at 38% for NAFLD and 14% for NASH. For diabetes, it was double that. So we're at 70% for NAFLD and we're at 35% for NASH.
And I remember Hispanic was over 50?
55% in Hispanics without other risk factors, just being Hispanic, and then about 25% with NASH. If you combine diabetes, obesity, Hispanic, we're talking about 90% and 45%.
One of the challenges for gastroenterology is they run a lot of procedure, and when we learned that they have a problem with time, because they spend a lot of time doing procedure, but VCTE is not particularly well reimbursed compared to some of the other things they could be doing. My immediate thought was, I asked, well, gee, what's well reimbursed? And everybody said scopes, and my immediate reaction was, well, San Antonio study, right?
When patients are coming to get their screening colonoscopy, typically they're fasting. You can offer them transient elastography to assess for their liver health, and you can actually be more targeted and just look at patients with type 2 diabetes or obesity, dyslipidemia. But these patients, once you identify that they have fatty liver disease, you can determine the severity of their disease and they become your liver patients as well. The other way that we do it actually is we offer transient elastography free of charge for anyone in the Phoenix and Tucson area. This is a program we started a couple of years ago. Patients come, we don't ask about insurance or anything, they get it absolutely for free. But if we identify fatty liver disease with significant fibrosis, we offer them to be seen in the clinic, and this way we have more patients walking through the door that become our patients. And we feel like this is one way to kind of identify early disease and give people peace of mind, because many patients will not have significant disease and you can reassure them. And then we identify about 20% that will have significant disease.
It's a fascinating way to build rapport with their patients and allow them to improve the quality of their life simply by learning more about what's not wrong with them.
Especially given the scarcity of transient elastography machines overall in the United States, we offer this to also gastroenterologists, primary care physicians, to send their patients to receive a free fiber scan, and then we send back the interpretation to the referring provider. And if they decide that they want them to see us in the hepatology clinic, we're happy to see them. But sometimes the GI provider just wants to know that elastography number, and we are happy to do it.
And now, back to Roger.
We hope you've enjoyed this recording. If you have any questions or comments about the content of this conversation or the entire episode, please send an email to questions@surfingnash.com. The next 3 weeks will be devoted to previewing and then covering the Paris NASH meeting and the EASL NAFLD Summit. September is a brutal month for travel schedules and, as a result, podcast scheduling. We'll be using some hybrid episode designs, and both Jörn and Louise will miss some episodes. I'll be here for all of them. It will be a challenge and a bit of a wild ride for me, but if you know me by now, you know I love a challenge. So stick with us as we move through the month. If you're in the U.S., enjoy Labor Day. Wherever you are, enjoy the transition to what we hope will be more temperate weather. Stay safe and surf on. We'll see you on the podcast. Bye-bye now.